All right, continuing off of our last where we left off, we're going to be talking about chromatin remodeling, histone modifications that promote chromatin condensation, attract histone modifying enzymes and other proteins that effectively promote and spread heterochromatin. So here you have heterochromatin specific histone tail modifications. So you have the heterochromatin, euchromatin, that's how it goes, and then between you have the barrier DNA sequence. Histone modifications attract heterochromatins to specific proteins, including histone modifying enzymes. So essentially, what we're seeing is in this whole thing here, right, where heterochromatins are found in nucleoli, and they are found in the centromeres and telomeres, whereas euchromatins are not found in the nucleoli. It's not dense, and it's actively transcribed. Um, Says less actively transcribed, but that is cut off. Now, what they're saying is that the histone modifying protein is essentially targeting the heterochromatin, because there's heterochromatin enzymes. These enzymes bind to this heterochromatin, and then the heterochromatin spreads until it encounters barrier DNA sequence. So then you have the decondensed chromatin, the histone acetylation, and then histone methylation. Right? You have the decondensed chromatin, you have the transcriptional activation, methyltransferases, it's giving you condensed chromatin, demethylation. HDACs, HATs, HMTs, HDMs. Honestly, I'm not really sure what's going on here, particularly, because they're talking about decondensed chromatin, right? And then transcriptional activation. So you have these decondensed chromatin DNA methylation, methyltransferases is making them into condensed chromatin, and demethylation is making them into decondensed, right? That kind of makes sense. And then you have histone acetylation, HDACs is making them into condensed chromatin, HATs are making it into decondensed, and then histone methylation, HMTs are what's turning it to condensed or trans, and then HDM is turning it into decondensed. So this is like an interesting table, I would say, and I would jot this down. So you have condensed, and then no, the first is decondensed, and this is condensed. So this is turning from DNA methylation right. So you have these three little packages, correct? And these three little packages are turning into condensed chromatin via methyltransferase, and then demethylation is what it's getting on the opposite direction. Condensed. And then histone acetylation. So you have all these little, again, you have all these little decondensed chromatin via HAT. You will get these decondensed chromatin. HAT is something that's funny to me. And then HDACs is what is making it condensed. Right? And then last but not the least, you have histone methylation, which is just, again, you have all these condensed, well, decondensed, pardon me, decondensed stuff, and that is turning into via HMTs and HDMS, it goes back and forth, right? So you have this is transcriptional activation, and then you have transcriptional repression. So repression is when it's condensed. It makes sense, right? You're condensing them, you're kind of putting them in a box, whereas decondensed is like they can be themselves. So that's like more um not repression. This the condensed one is repression, the other one's like transcriptional activation. That's transcriptional repression when you're condensing it. I was trying to remember in some sort of mnemonic because you have the methyl MD MDHHHH MDHHHH is how I would remember it. So you have methyl transferase, demethyl transferase, HDACs, HD like the TV, HDAC, TV and AC, right? TVAC. So you have MD. If you're an MD, you get to buy TVs and ACs, and then you have the hat. You also get to buy a hat, TV AC hat, and then you get to buy HMTs, H&M clothes, right? HMTs, and then HDMI cables. So how I'd remember this is this: once you become an MD, you you get to buy TVs, you get to buy ACs, you get to buy hats. You also get to buy HMTs, and then HDMI cables. Hopefully, this is one way you could remember. So I would try to write this down somewhere. Sorry. So MD, demethylation, methyl transferase, MD, and then HHHH. So once you become an MD, you get to buy HDTVs, right? HDACs, hats, um, HN something, and then the last one was HDMS. So HN is the only HMTs, HMT, H and M, right? H and M. It's not HN. So HMTs. So that's how I would remember it. So you have MDHHHH, methyl transferase, demethylation, trans HDAC, hats, no, HDAC hats, HMTs, and HDMS. That's it. That's how you do it, folks. So mechanisms of chromatin remodeling too, we're talking about chromatin remodeling complexes. They contain enzymes that catalyze ATP hydrolysis and use energy thus produced to force DNA movement along histone octamers. So essentially they need ATP. And how does this work? These chromatin remodeling complexes, right? They are the ones pushing the enzymes um, that catalyze ATP. So ATP needs to be broken down. Um, so the chromatin remodeling complexes have these enzymes, and they are the ones that produce this ATP and use this or catalyze breaks down the ATP to give energy, and hence DNA movement is forced along the octamer. So these chromatin remodeling complexes Remodeling complex has enzymes breaking down ATP to ADP to get energy to move DNA along histone octamer. Then you have chromosome X inactivation. So cells in the early embryo, you have two X's. These inactivation of randomly selected X chromosomes. So if you inactivate one of these X chromosomes, you directly inherit the pattern of the X chromosome inactivation. So it's just being what it's saying is that essentially if you inactivate one, it multiplies and multiplies again, and that inactivation kind of stays. Um, Depending on which X chromosome has been inactivated. This inactivated X chromosome, bar body, and then you have the region containing the active X chromosome not visible. So I would quickly go over this with my professor. So this is 17, right? 17. And it's inactivation. Then you have the genomes. A genome is the totality of genetic information belonging to a cell or organisms, in particular the DNA that carries this information. Genomes vary across the tree of life. The genome of viruses are the most variable. They can be made of RNA or DNA, are either double or single-stranded, because RNAs are obviously single-stranded, whereas DNA is double-stranded. Here it's a little blurry, but we're talking about the RNA viruses here, and this is talking about the DNA virus. DNA viruses include herpesvirus, adenovirus, and papillomavirus. And then you have, for RNA virus, you have the Ebola virus, the rabies virus, the mumps virus, the HIV. So there's more variety to RNA virus. I'm thinking because it's smaller than there is for DNA. And then prokaryotic and eukaryotic genomes are always double-stranded. DNA, right? So you have the prokaryotic here. Prokaryotic and eukaryotic are always double-stranded DNA. So it doesn't matter which genome it is, it's always double-stranded. It doesn't matter whether it's prokaryotic or eukaryotic. So genome is essentially the totality of genetic information, not just the gene itself, but the total thing. Remember, all the way up here, when we were talking about how, see, this is the whole genome. And within the genome, there's coding RNAs and transcriptome, there's coding RNAs, there's non-coding RNAs. So the genome is essentially the totality of the gene rather than just parts of a gene. The totality of a genetic information belonging to a cell or an organism, in particular the DNA that carries this information. Human history traced by analyzing genomes. Region inhabited in Netherlands, interbreeding humans, Neanderthals. Yada yada yada. I don't really know what's going on here. I'd have to consult the video again. 19 human history. Not important. Important question mark. Then we are going over the typical difference between any human beings. You have the single nucleotide variations, the size of a nucleotide pairs. Difference might be 3 to 4 million. Small deletion. There's a lot. So essentially, what they're trying to say is there's a lot of differences between one human being's genome sequence in reference to human genome. The low complexity, simple sequences repeats, yada yada yada. Mobile element insertion, structural variation, karyotypical. No.